today's lecture is probably going to be short. Yeah, because we're going to just cover materials for amino acids, right? We talked about how different amino acids are, right, compared to glucose and fatty acids. Okay, the difference is in this amino group, right, that has to be removed when it is being catalyzed. We don't see this amino group in glucose or fatty acids, right? They are all mostly just carbon and hydrogen. That's why we call them hydrocarbons, organic fuel. So when you see the word organic, it means there's a lot of carbons and hydrogens and oxygens. So, but at the same time, the amino acids have carbon skeletons, right? They do have the carbon skeletons that we can harvest for energy. Okay, so we need a balanced diet from carbohydrates, fats, and proteins. Proteins will give us amino acids. Right? So the amino acids will, the carbon skeletons are derived when the amino acids lose the amino group, right? And we call them alpha keto acids. Right? We'll look at the structure of alpha keto acids, and they can eventually be completely oxidized again to our final product, carbon dioxide and water. Right? And in the process, we harvest the energy in terms of ATP by sending these units, by sending these alpha keto acids into the citric acid cycle. So everything converges into the citric acid cycle. Glucose, fatty acids, and amino acids. Everything goes through the citric acid cycle. Okay? And and we will notice that the amino acid, the ammonia or ammonium ion has to be removed, right? It is also useful for biosynthesis for amino acids or nucleotides, right? Remember, nucleotides is the other organic molecule with the nitrogens and biological amines, right? But if there is excess from these processes, it's not required for biosynthesis, it has to be removed from the body because excess amino acids cannot be stored, right? In its free form, okay? Excess amino acids cannot be stored. So what do? We send it into the urea cycle. The urea, okay, which is the excretion product, will be removed in the urine. Why do we do that? Because ammonia is toxic. We can't just let ammonia accumulate in our bloodstream, circulate. Right? It's toxic and it's alkaline, it changes pH balance, it's quite destabilizing. So we change it to a non-toxic form called urea, right? Remove it in the urine. And these two cycles are linked, right, in what we call Krebs bicycle or known as the shunt, aspartate argininosuccinic shunt, that links up these two. Okay, some of the byproducts from the urea cycle can be fed into the citric acid cycle. This is just to for the economy of the system, so that you don't waste any single material. Okay. So we talk about metabolism of amino acids mainly occur, mostly occur in the liver. Okay, liver, very rarely elsewhere. Everything is happening in the liver. Okay, so in the liver, they go through on the chemical process or the enzymatic process called transamination. Right? Transamination is a very important step in this transport of amino acids to the liver. Right? So the form that transport of the amino group. So the amino group from all these amino acids are carried, carried by glutamate finally in the liver. Okay, this is the final nitrogen carrier transporter, okay, glutamate. So in the liver, if it's present as a glutamate, the glutamate will go through deamination, okay, and transamination to allow the the ammonia group to be removed as urea. Okay, this is the urea cycle. We'll look at it in detail. But elsewhere, especially in the muscles, the excess amino group is not carried as glutamate. It is carried instead as alanine. Okay, alanine. So because we said that the muscles usually respire anaerobically. They go through the glycolysis cycle, right? They produce pyruvate, right? This is glycolysis, right? Like glucose, glycogen will become pyruvate to supply the energy and allow muscle contraction. And the amino acid will undergo, any excess amino acid will undergo transamination by passing the amino group to pyruvate. Right? Because pyruvate is readily available there. And pyruvate becomes alanine. Right? Alanine goes into the blood and is transported to the liver. Right? Right? Why do we do that? Did you see it goes back in a cycle? So alanine passes to alpha-ketoglutarate to become glutamate, the final carrier form, and then it goes back to become pyruvate. All right, in this what we call glucose-alanine cycle. Right, so this is again the body's clever way of economy, fuel economy. You can recycle. Right, this is recycling. This is true recycling of your materials. Right, you can remove waste at the same time. Take your bring your transporter back into the muscles to continue to allow to feed the muscles fuel for contraction continually. All right, so you have to appreciate the body is such an amazing machine. Right? Everything just works in such a nice coordinated fashion. Right, everything's all linked up. Right. Okay. So then in other tissues. It's not carried as alanine, but carried as glutamine, right? So glutamine, probably because pyruvate is not in excess, and then glutamine will pass to the liver and then will be passed to the liver and then to the mitochondria again to become glutamate. Right? Glutamate and glutamate is very similar, right? So you have to know the similarity between the structures of the amino acids. Okay, so final form, glutamate. Right? Then we talk about transamination. This transamination process is different from deamination. Completely different. Okay? Trans and de. You can get some idea from the name trans. Trans means transfer. Right? De means remove. Okay? So in this transamination process, which is involved in transport of amino group to the liver. Alright? It is not removal yet. Okay? It is the transport to the liver. The amino group is like pass the parcel. Okay, we pass the parcel from one molecule to another molecule, and it's catalyzed by enzymes called aminotransferases or transaminases. Same, okay, same. And what is special about this aminotransferases is that they use this coenzyme called pyridoxal phosphate. Right, we saw later on, yeah. And the whole scheme is as such. The amino group is finally transferred to the alpha carbon of alpha-ketoglutarate, and this becomes glutamate. Remember, glutamate is the final one. Alpha amino group gets transferred to alpha carbon of alpha-ketoglutarate, right? And then the amino acid leaves as an alpha-keto acid. Okay, so the purpose really is to collect as many amino groups as possible in the form of L-glutamate for removal. Okay, glutamate is the amino group donor. Okay, at the end. Yeah. So then we look at the coenzyme pyridoxal phosphate. You will always see this with any transaminases, any aminotransferases. It is a vitamin B6, like most of your coenzymes, right? Your NAD, your CoA, your FAD, etc. They are usually vitamin Bs. Yeah? Some conjugates of vitamin Bs. In this case, the vitamin B6 or pyridoxine, it looks like that. Don't need to remember the structure, don't worry, just understand what it is doing. So in this coenzyme, right, the coenzyme associates with the enzyme. How? How does your coenzyme associate with the enzyme? Right? It could be, it's sitting somewhere else, not the active site. That's the idea of coenzyme. And it interacts with by non-covalent interactions. Okay, non-covalent interactions like electrostatic interactions, hydrogen bonding, van der Waals, etc. But in this case, for pyridoxal phosphate, it also interacts with the enzyme via a lysine residue. This lysine residue and the pyridoxal phosphate forms what we call a shift base. So the shift base looks like that. It's basically a CN double bond. Alright, a CN double bond with R groups all around. Okay, if you see this kind of structure, it's called a shift base. Or you will see the chemical name imine, I-M-I-N-E, right? All over the place. Okay? So in this case, the OH group is involved. It has, this is the vitamin, and the phosphate version, okay? Phosphate version of the vitamin will be the coenzyme. And again, you see the 5' is phosphorylated. Okay? Phosphorylation will always occur with the hydroxyl end, okay? Phosphorylated, and then this hydroxyl group becomes an aldehyde, and the aldehyde is the prelude to the imine formation, to the shift base formation. So again, if you look at this case, that's why I say don't need to memorize structure. Just memorize this portion. It could be very complicated, but once you see this, it is a shift base. Okay. So CN double bond with R groups. And this is the enzyme. This is a lysine residue, right? Does it make sense? Because the lysine residue has N groups on it. Okay? Lysine has N groups on it. So the lysine residue on the enzyme will bind to the coenzyme using a shift base. At the same time, there are other non-covalent interactions elsewhere, especially electrostatic interactions with the enzymes. The enzyme is huge. It could be all over. Okay? So this shift base can undergo reaction to form a primary amine on this group. Okay? And then this is how it is then used to carry amino acids from incoming amino acids. Let's look at this scheme, right? So imagine the beginning, right in the beginning. The coenzyme is sitting on the transaminase, and it is linked up with all the electrostatic interactions and at the lysine position. Okay? There's a shift base formation at the lysine position of the enzyme. Then there is an incoming free amino acid that you are supposed to remove, okay? From which you are supposed to remove the amino group. Okay? So what does it do? The amino acid will transfer its amino group to the paradoxal phosphate. So naturally, the paradoxal phosphate will have to disrupt its interaction with the lysine residue. Okay? But at the same time, it is still bound to the enzyme through other non-covalent interactions. Okay? So you have, when it is being passed, the whole amino acid, right, is being passed onto the CH residue, right, on the paradoxal phosphate to form a shift base. Again, C double bond N, shift base. And this whole moiety is the amino acid. Okay? CNH2, COO minus, and R. So this is amino acid. Then it undergoes rearrangement, right? Undergoes rearrangement eventually to release, because all we want is the nitrogen on the paradoxal phosphate. We don't want the alpha keto acid. So the alpha keto acid has to be hydrolyzed. Okay, so water is here, hydrolyzed, and this is what we call alpha keto acid. Why do we call alpha keto acid? See, alpha. Carbon is alpha to the carboxylic group, right? And this is a keto group, right? And this is an acid group. So alpha keto acid, right? And this could be any R, okay? Could be any R of the amino acid. So once hydrolysis happens on the coenzyme, the amino acid leaves as an alpha keto acid, and look, the amino group is finally transferred, right? Transferred to the coenzyme over here, and it becomes a paradoxamine form of the coenzyme. Okay, see, it's being passed, right? So then it is just an intermediary molecule. You cannot hold the amine group the whole time. It has to become glutamate finally at the end, right? So then we need another molecule or transport molecule in the form of alpha keto acid, which is, in this case, alpha ketoglutarate, right? Only alpha ketoglutarate can become glutamate because their structure is very similar, okay? And you just need to transfer the amine group to alpha ketoglutarate, it will become glutamate. So alpha ketoglutarate then comes in, right, diffuses near the enzyme, and it becomes bound, okay, to the coenzyme the same way. You see the reaction, right? So this is a reaction. It's actually called a ping pong reaction. In enzymatic kind of mechanism, it's a ping-pong reaction. It's going back and forth, right? Pass, pass, pass. Okay, this is a ping-pong reaction. So now the ping is here, the pong comes back, yeah? And then over here, this is alpha-ketoglutarate bound to the amine group. Again, rearrangement. And then this product now looks back. Again, like an amino acid. And now in this case, the amino acid is glutamate. Right? Glutamate is our final donor, right, in the urea cycle. So this product too is glutamate. Right? And because the whole thing leaves, this coenzyme is regenerated. Okay? Regenerated. If it's free, it will then bind to lysine residue on the enzyme. If it's not free, if there's another incoming amino acid, then the whole cycle repeats itself. Okay? Right? So I hope this is clearer now. So this whole cycle will go on as a transfer reaction. Again, I mentioned that there is no net loss of amino group, therefore transamination. This is just an extra slide to show you the similarity between alpha-ketoglutarate and glutamate. Yeah, again you see COO minus CH2, CH2, CO. So alpha keto acid. And then if you hydrolyze this and then do transamination, you add an NH3 here, it becomes glutamate. Okay? Alright? So for the amino acid, the amino group that has been passed on here will become an alpha keto acid. Right? So all the transaminases will use pyridoxal phosphate as the cofactor. Okay? So what happens to the glutamate in the liver? Right? It is still not the final step. Right? In this case, imagine all your excess amino groups is now collected in the liver. Right? It's your garbage center. Right? Your amino group in glutamate. So in the hepatocytes, the glutamate is transported from the cytosol into the mitochondria. Right? This amino acid metabolism is the one that straddles between cytosol and mitochondria. Right? The rest is all quite compartmentalized. So glutamate is transported from the cytosol to the mitochondria, where it undergoes now oxidative deamination. Now it's a different mechanism now. It's deamination. So we have to remove the amino group from the glutamate. And this is catalyzed by another enzyme called L-glutamate dehydrogenase. And what is special about dehydrogenase is that this can use either NAD or NADP as the oxidizing agent. So previously in the fatty acid oxidation, we said that NADP is usually present right in the reduced form, whereas NAD is always present in oxidized form in the body. But in this case, this is an exception. Okay? Glutamate dehydrogenase can use both in this oxidized form. Right? Okay? So we changed this slide. I made a mistake by writing transamination on top. So if you could change it, it becomes oxidative deamination. Right? So the name gives you a clue. Oxidative means there will be all these electron carriers or reducing agents, oxidizing agents involved, and removal of amino groups. So we look at glutamate structure again. Glutamate has two COOH, right? Okay, so objective is to remove this amino acid. Okay, you can use NAD or NADP, right? And then it has an intermediate imine group, and finally becomes alpha-ketoglutarate. Alright? Okay. So now, because it's a removal process, the deamination process, you get a free ammonia here. It's not transferred to any molecule, it's free. This ammonia is free. Okay, free. Therefore, deamination process. So the enzyme should be changed to L-glutamate dehydrogenase over here. So what happens now? Okay, so of course this is quite simple. Alpha-ketoglutarate can always, where does it go? Not wasted. Again, nothing is wasted. Where does this alpha-ketoglutarate go? It can go back into the citric acid cycle, right? The problem is ammonia, where does it go? Okay, you can go to biosynthesis, make more nucleotides, but how much nucleotides can you make, right? Right, so especially if you eat a lot of proteins, if you eat a lot of like huge, nice steak meal, you go to Morton's or Lorry's or any steakhouses, right? You have a lot of amines and ammonia circulating in your body. Okay, so now in the hepatocyte, right? This is the process. It doesn't show the urea cycle yet because we need to process. We need to process the ammonia. Okay, we need to process the ammonia over here. So just now we stopped here. Glutamate, by glutamate dehydrogenase becomes ammonia here. Right? So the very first step, and this is what we have seen, right? Alanine to pyruvate. Okay. So over here, the ammonia becomes a molecule called carbomoyl phosphate, and ATP is needed. All right, let's take a look. Yeah, what happens? Okay. This is what I've mentioned about the glucose-alanine cycle, right? Okay. Just now we had the same picture without the muscles here. Okay. So you have again instead of glutamate in the muscles, it is being transported as alanine, and then into the blood. So blood alanine, and then alanine into the liver, and liver alanine passes the amine group to alpha ketoglutarate, and this is alanine aminotransferase. Okay, so the way to name it is always to name it from by the donor, alanine aminotransferase, right? If this is aspartate, then it will be aspartate aminotransferase. Okay, so it becomes pyruvate, and pyruvate undergoes this process called gluconeogenesis to make glucose in the liver, and then back again. Okay, again, why do we do that? Because only the liver can perform this function called gluconeogenesis to make glucose. You have to spare your muscles to do something else. You cannot make the muscles do gluconeogenesis. Then they cannot focus on contracting, right? So the body kind of compartmentalizes the function, right? So the liver is where I would send everything to be made, and I focus on moving around, right? So that's why the pyruvate, that's why we use alanine instead of glutamate, because pyruvate and alanine structure is hugely similar. So by just a very simple amino transfer reaction, I can regenerate my pyruvate in the liver, which is a raw product for glucose synthesis. Okay, so you can't do that with glutamate, right? And then this glucose gets sent back into the blood and then back into the muscles again to continue, to allow the muscles to continue contraction. Okay, all right. So this goes on. So now we're at urea cycle. So we will see what is happening in the urea cycle is discovered again by Hans Krebs, the same guy, citric acid guy. Okay, Krebs guy. So now where are we now? We are in the liver. Okay, we're still in the liver, and where are we now? Cytosol or mitochondria? Mitochondria. Okay, so now we're in the liver. Mitochondria of the liver. And what are we now? Ammonia, okay? Because the glutamate has already been deaminated and we are left with ammonia. And then we said, okay, we cannot remove ammonia in its form like this because it is toxic, right? In high concentration, it causes brain swelling, right? And besides, it is very permeable. It passes through membranes and they raise the pH of acidic compartment of cells. This is not good. Why? Right? And the cells deal with foreign materials by acidifying them. All your immune system works that way. Even in the cells, phagocytosis, endocytosis, okay, there is always this acidification function to kind of dissolve everything else, right? The viruses are being killed that way, and foreign materials are being killed that way. So we cannot allow anything to change the pH of this acidic compartment, right? That would be dangerous for us, okay? So it's a protective mechanism. So therefore, this ammonia has to be converted. And of course, brain swelling is not acceptable. Okay, so we convert it to urea. Why to urea? Because it is neutral, neither basic or acidic, and it also has a simple function, a function in the kidney as an osmolyte in small amounts, okay, to reabsorb water and useful ions. When you go to your higher year 3, year 4 therapeutics, then you will start seeing these parameters. When you do blood tests, urea, okay, blood urea nitrogen, or it's always like or creatinine is always a parameter that you look up for in the chemistry, okay, for the patient, right? And that's because it has an additional function in the kidney. It can help you look at the kidney function of the patient so that you know how to dose your drugs. Because you know the dosages of drugs is affected by hepatic function and kidney function. All right, so you will learn that in year 3, year 4. But so in this case, if it's not used as osmolyte, then it is just being removed in the urine. Okay, in the urine. This is the similarity in structure. You have ammonia, which is very simple. Then you can understand how come it can pass through membrane so easily, because so small and permeable. This is urea. Not difficult to remember, right? Look, it is symmetrical. Okay, symmetrical with the carbonyl group in the middle, left and right amino group. You see, if we just now transfer of ammonia group is only one molecule, but over here you can carry two. You can carry 2 molecules in your urea. Right? So from a motorbike you have converted into a car. You can carry more passengers, okay, in this urea molecule. So that's the function, right? Increase the capacity of removal. And over here is uric acid. This is usually for reptiles, right? For reptiles, this is how they remove the nitrogen in the form of uric acid. And also in patients, I think Dr. Ho has mentioned, uric acid also causes this condition called gout, right? Accumulation in the joints, right? Because why? It is not very soluble. It becomes crystals in your joint, right? And it causes pain in the joints. All right, so urea is our preferred form. The very first step, this is the overall reaction of the ureal cycle. Okay, the overall reaction. So you have ammonia, and in this case, you need help from bicarbonate. Where do we get this bicarbonate? Where do we get this bicarbonate? It is simply carbon dioxide, okay, dissolving into the cytosol in the aqueous condition, and it will form bicarb. Right? And where do we get the carbon dioxide from? In the mitochondria, you have citric acid cycle. Right? Carbon dioxide is formed. So you get the carbon dioxide from the citric acid cycle in the normal respiration. And over here, there is another, this is the overall reaction. So in the overall reaction, there is involvement of aspartate in the final step. Okay, in the final step, whereby you have another amine group being added. So this is the green color, matches the ammonia, okay, derived from glutamate. Red color is derived from aspartate to make our urea molecule. Okay, so one molecule from ammonia, one molecule from aspartate. Right. When this aspartate is removed, when the amine group is removed, aspartate becomes fumarate. Again, fumarate. It is an intermediate in the citric acid cycle, and this is recycled. Okay. This is the general reaction. Okay, we will go through it stepwise. And some of the mechanics of it, urea production occurs almost exclusively in the liver. I've been repeating that. And it begins in the mitochondria, but there are 3 other steps that is in the cytosol. Right? And finally, urea travels in the bloodstream and to the kidneys and removed as urine. Okay? So this blue thing is the mitochondria. This blue compartment is the mitochondria. Alright? And this portion is in the cytosol. So cytosol over here. Okay? This reaction is in the mitochondria. Okay? The glutamate dehydrogenase, although it look like it's out of the mitochondria, but it is in the mitochondria. Okay, so this is in the mitochondria. Okay, so let's look at this part here now. Okay, this part here is the urea cycle. Enlarge for your reading pleasure. So, carbon phosphate, step 1. So we said our raw materials, in beginning ingredients, ammonia, bicarb, and then it has to become carbon phosphate. Right? So the bicarb is over here, HCO3, right? And the HCO3 needs the help of an ATP, okay, to hydrolyze ATP. And ATP becomes ADP. So it is also kind of like a coupled reaction, yeah? And becomes a phosphate, a carboxyphosphate over here, right? And this carboxyphosphate is the reactive intermediate, which can then combine with the ammonia over here. And phosphate group is removed as the inorganic phosphate over here to become a carbamate. Okay, so NH2. Right, and the COO minus. It's a very simple structure with two functional groups, carbamate. And then another molecule of ATP comes in to become carbamoyl phosphate. Okay, so the carbamoyl is derived from the carbamate, which is the COO and the NH2, and then the phosphate group. It donates its phosphate group into this molecule. Okay, this reaction is catalyzed by an enzyme called carbamoyl phosphate synthetase 1, and this is found in the mitochondria. There is another isoform called carbamoyl phosphate synthetase 2 that is found in the cytosol, and we'll see it on Monday for nucleotide metabolism. Alright, so don't get both of them confused. So in this case, it is a mitochondrial version, and it's synthetase 1. Right? So the enzyme uses 2 ATP and bicarbonate this process. And so where did the ammonia come from? It can come from the amination of glutamate, which is the collecting molecule, or sometimes it could also come from intestines, right? We have bacteria in our stomach and intestines, and they could oxidize our amino acids that's been digested from our food directly. And this will be in the bloodstream and collected to the hepatic portal vein, and then it drains into the liver. Right? This is the first pass. So after it is being made into a carbamoyl phosphate over here, it then goes into a cycle. So this is the second cycle that we have seen already. The first cycle is the citric acid cycle, and largely similar, only that the intermediates are changed now. Okay, so we have the carbamoyl phosphate coming in, and in the citric acid cycle is the acetyl OA. Both are high energy compounds, right? So both are high energy compounds. So over here, it is going to be combined with this molecule called citrulline. And let's look at what a molecule called ornithine to become citrulline. And if you see these yellow circles here, what do you think these yellow circles are? What are the yellow circles? Do you remember what is the problem with the mitochondrial membrane? Yeah, very impermeable, right? So these are transporters. Okay, because urea cycle straddles between the two compartments, right? You have to get it out. And in order to get it out, you can't let it diffuse out, no way. So the substance have to be transported through a pore, a channel, out, okay, of the membrane, a gate to open it out to the cytosol. So these are all transporters. Yeah. So let us look at ornithine. So ornithine, so in citric acid cycle, we have oxaloacetate, right, coming in to become, and then becomes citrate. Over here, ornithine combines with carbamoyl phosphate to become citrulline, and this reaction is catalyzed by ornithine trans-carbamolase. I didn't put reactions here because I don't think it's necessary for you to memorize the structure, but for you to look at the difference. Okay, so maybe this will help you remember it. And let's see. Okay, so look at ornithine. Ornithine is almost like a lysine. So in the end, you just have to remember amino acids, right? Lysine, it looks like that, right? Okay, NH2, and this is the R group. R group, okay? And ornithine is exactly almost like lysine, but only a carbon atom shorter, CH2 shorter. Okay, all right. So if you count, this is a carbon atom shorter. And the product, this is the product citrulline is almost like an arginine. The arginine looks like that, like with two amino groups over here, right? And then the NH here. But the R group has a urea group at the end. Remember, your urea is symmetrical. CO, NH2, NH2. So citrulline is an arginine with an urea group at the end. So the cycle already gets it ready, right? Cycle already gets it ready to be cleaved off as a urea molecule, and then recycle the rest of the molecule, the carbon skeleton. So in this case, ornithine will combine with carbonyl phosphate. Phosphate is gone, and then it becomes a citrulline molecule. Okay, over at this end. Right, this end is untouched. Okay, this end is untouched. The reactions are at this end. And finally, citrulline is then exported via the transporter to the cytosol. And in this case, an unusual reaction takes place. Usually we have seen a nucleophile attacking a carbonyl carbon over here, right? But in this case, somehow, the carbonyl oxygen acts as a nucleophile. Yeah, this doesn't occur very often. I've only seen this in this reaction. So citrulline, this is the arginine moiety, but instead of arginine, you have urea at the end. All right, so objective is to cleave off this, but we need intermediate. So this carbonyl oxygen, right, acts as a nucleophile, right, and then attacks the alpha phosphate group of ATP. So ATP, we have alpha, beta, gamma, right? So it goes straight in for the alpha phosphate group. And at the end of it, you have you need aspartate to come in over here to make a very huge molecule, right? This second step. So first step is AMP is linked to the oxygen here, the urea oxygen, and then aspartate, right, with the nitrogen atom. This is what we usually see, the nitrogen as a nucleophile attacking the carbonyl carbon, all right? Okay, into the C here. And then you have this big molecule called argininosuccinate. Right, this is the arginino. See this arginine molecule, arginine structure over here, right? NHC, NH2, and this is a succinate molecule, right? Succinate is also symmetrical, right? With two COO minus, CH2CH. Okay, so this intermediate is a huge molecule called argininosuccinate. Right, and the intermediate will then undergo, right, elimination reaction, right, to become fumarate and arginine, right? Okay, so this elimination reaction. So when I put it like that, means I don't need you to memorize the structures. Yeah, the elimination reaction. Yeah, the proton is abstracted. And a C double bond is made. So instead of a succinate, we get a fumarate because the double bond is being made. Right? Over here is a succinate, but now we put a double bond in here, right? And it becomes a fumarate at the end. And then release the arginine. Okay? Right? So arginine plus a fumarate. And it's catalyzed by argininosuccinase. And the final step, we have the arginine, right? Arginine plus water. You become urea and ornithine. Okay? Urea and ornithine. In the beginning, it was nitrogen and ornithine, right? But at the end of the reaction, you get urea and ornithine because you need to feed the ornithine back into the cycle again. So you have to regenerate the ornithine. Okay, so this reaction is catalyzed by arginase. The cycle is considered complete and the ornithine is recycled. Okay, so over here, okay, citrulline and this huge molecule argininosuccinate, right? And succinate becomes a fumarate, and arginine is removed. Okay, so just this intermediate molecule cleaned off. This is arginine. And fumarate, because the succinate becomes reduced, it becomes a double bond. Right, arginine is removed, and the arginine is hydrolyzed. Okay, it's hydrolyzed over here to become urea and ornithine. So the fragment is just being defragmented over here. Okay, arginine, okay, is hydrolyzed to become urea and ornithine. Ornithine goes back to the mitochondria. Alright, so it's more straightforward than the Krebs cycle. So the fumarate in this case, okay, as we mentioned, it can go back to the Krebs cycle. Right? Within the liver itself, in the mitochondria, the Krebs cycle is ongoing anyway. Right? Everything is going on at the same time. You have the urea cycle, you have the Krebs cycle going on at the same time in the mitochondria, because all the enzymes are there. And so fumarate can easily participate in the Krebs cycle reaction, or it can also, in times of starvation, or in times of low glucose, undergo gluconeogenesis by converting it to a malate. Right? These are intermediates for your citric acid cycle, which are required to remember anyway. So malate, and then it becomes an oxaloacetate. This is the last part of the cycle, isn't it? This is the end bit of the citric acid cycle. So instead of letting oxaloacetate go back into the cycle and get combined with acetyl-CoA, it will then be sent to make glucose in gluconeogenesis. Okay? So this Krebs cycle link that we were talking about, Krebs bicycle, is linked through a shunt. Just now we saw it's called the aspartate arginino succinate shunt. And the shunt is there because of this fumarate molecule. Right? So we have fumarate at the end. So we said that it's transported to, converted into malate in the cytosol, and then transported to the mitochondria. Okay, and the mitochondria, fumarate is over here. Okay, you see it going back to the Krebs cycle over here. Okay, and then we describe this process from fumarate to malate to oxaloacetate. Then it goes round and round to make ATP. Okay, or there's an alternative pathway for fumarate to become malate directly in the cytosol, and then goes back into mitochondria to become malate. So you see, there are two pools of fumarate and malate, and two pools of enzymes inside the cytosol and in the mitochondrial matrix. Okay, and the aspartate form in the mitochondria via transamination will become the nitrogen donor, okay, in step 3. Okay, so oxaloacetate and glutamate can also undergo transamination to make aspartate, right? And aspartate will then be fed into the cycle. So this is the connection, okay? You see that urea cycle produces fumarate, which then goes back to the citric acid cycle. This is the bicycle, right? You see, if you pedal here, fumarate goes into the other wheel and power it, right? On the other hand, this wheel can make aspartate via oxaloacetate and glutamate, and then sends the aspartate to the other wheel to power it to be combined, right? To form urea at the end of it. Okay, that's how the connection goes. The materials fed into each other to move the wheels, okay, continuously. And this is possible because of a physical, because of a shunt, right? It's not a physical shunt, but it's a chemical shunt, a chemical connection, a chemical link between these molecules. Okay, so we call it the aspartate arginino succinate shunt, because the connection occurs when we make this arginino succinate in this step. Okay, So this is the argininosuccinate step. The aspartate is over here. Okay, participate in this step. Right? Therefore, we call it the aspartate argininosuccinate shunt. This is the shunt reaction. Okay, again with the cycles, we look at regulation. Okay. The regulation of the urea cycle is on a long-term basis is regulated by your diet intake. Right? If your diet is rich in proteins, then the amount, the intake will then dictate, right, how active the enzymes will work. But on a short-term basis, right, on a short-term basis, the urea cycle can be regulated by arginine allosterically. Okay, and this is how it is done. Okay, so the arginine will stimulate this special enzyme called the N-acetyl-glutamate synthase, okay, to make an intermediate called N-acetyl-glutamate. Now, this molecule exists only as a purely signaling molecule for this regulation. Okay, this N-acetyl-glutamate, okay, it's made purely for this purpose. Right? It is not doesn't have any other biological functions. Right? It is not a raw material or anything else. Okay, this combination, which is catalyzed by the enzyme called N-acetyl-glutamate synthase, right, in response to high arginine, which is i.e. high amino acid removal required, to make this N-acetyl-glutamate, which will then inhibit the carbamoyl phosphate synthetase 1. Okay, this is the very first step. The first step of the cycle whereby ammonia and bicarb combines, right, to give you the carbamoyl phosphate. Okay, so usually when you have a rich protein diet, the rich protein diet could reduce the enzymes. So for people with a low protein diet, usually all the urea cycle enzymes are very low. Okay, very low. The amount is very low. But for high protein diet person, right, or in terms of starvation, right, okay, then this would set into motion, right, because during starvation all your stores are being mobilized. Proteins is even broken down to mobilize to become glucose. So the action, the regulation will come in. So on a short-term basis, the body produces this N-acetyl-glutamate by the synthase, right, and it inhibits it right at the beginning, where it is synthesizing, where it inhibits carbamoyl phosphate synthetase 1 to stop the production of carbamoyl phosphate. So nothing will be fed into the urea cycle. Okay? Sorry, this should be stimulating, okay, not inhibiting, stimulate, right, stimulating. Okay, the N-acetyl-glutamate stimulates, okay, this synthetase or removal. Yeah, so it's not inhibiting, it's stimulating. Okay, more bicycles for you, right? So you have learned in your amino acid lectures that you organize your amino acids by chemical structure, polar, non-polar, hydrophobic. Okay, but today, after the metabolism class, you will learn another organization of amino acids. And this organization is based on the metabolism, based on the anabolic and catabolic reaction. Okay? In this case, what am I trying to tell you? Okay, you don't ask me if you have to memorize, answer is no. But I just want to impress on you how the amino acids connects both the anabolic reaction and the catabolic reaction of citric acid cycle. You see the amino acids being pointing down and up, see? Okay, it could either be fed into the catabolic reaction, or it could be, intermediates could be taken out to make the amino acids. Right? And based on this picture, this is a better picture actually, yeah? We can tell that amino acids, the other classification of amino acids is based on whether they are ketogenic or glucogenic. Okay, what do we mean by that? So we have not really talked about anabolism, right? We have no time to talk about anabolism. So we're only focused on amino acid catabolism, right? The removal. And in this case, ketogenic amino acids are degraded entirely or in part to acetyl-CoA or acetyl-acetyl-CoA. This one we have seen, right? In beta oxidation, formation of ketone bodies. And this is here. The blue parts. Blue parts are ketogenic. Okay, so this it occurs during starvation. Same thing as beta oxidation, starvation, all right, or diabetic patient where the glucose is not well controlled. Then the body has to mobilize all the other forms of materials in the body to make energy. So same thing, the proteins in the body, proteins in the muscles, yeah, will be broken down, which is not good news, and we try to avoid that. To make amino acids. To break it down the amino acids. And these amino acids will then become acetyl-acetyl-CoA or acetyl-CoA to form what we have learned as ketone bodies. Right? Finally, ketone bodies will go to different parts of the body to become fuel, to be fed into the citric acid cycle back again. And we have, so these are the ketogenic ones. Right? On the other hand, we have the glucogenic amino acids, which are converted to glucose, glycogen via glucogenesis. These are also powering the system, but in the end, they make intermediates. These amino acids can make intermediates that go directly into gluconeogenesis process. Okay, so they are the pink one. All right. So the molecules that are involved in gluconeogenesis are your succinyl-CoA, your fumarate, and your oxaloacetate, and even your pyruvate. Right? That's why you see the amino acids feeding in, right? Because they form the carbon. We make use of the carbon skeleton to make these intermediates, which will then become glucose or glycogen via glucogenesis. So glucogenic. And out of these, we have 5, which are, well, the definition is quite hazy, right? 5 of them are both glucogenic and ketogenic, right? You see them in both, okay? Which is over here, isoleucine, threonine, tryptophan, phenylalanine, and which one? 1, 2, 3, 4, 5. Tyrosine, okay? Okay, so you have 5 of them which are both ketogenic and glucogenic, overlapping. Okay, so this is for you to appreciate. Okay, the fact that amino acids is mobilized differently. So even though they are broken down, we know how the ammonia group is removed. The carbon skeleton are potentially useful, right, to be harvested as energy. We don't have time to go into the mechanics of it. Right, and finally, in anabolism, just one slide to summarize it. So you've learned in your lecture that amino acids are divided into essential and non-essential, right? Besides the chemical definition, polar, non-polar, we also have essential and non-essential, right? So what do we mean by essential amino acids? Do you still remember? What do we mean by essential amino acids? Can we make it or can we not make it? Can we synthesize or can we not synthesize? Essential means we cannot synthesize it de novo in our body. Okay? We cannot synthesize it from simple molecules in our body. Therefore, it is essential that we take it in from our diet. Right? So this is essential. Non-essential, on the other hand, can be produced by the body. Okay? Yeah? So in plants and bacteria, they can synthesize all the 20 amino acids, and this cycle is for the plants and the bacteria. Okay? And again, you see that it's being connected to the central axis of the glycolysis and the Krebs cycle, right? From the materials that can be taken out, these intermediates from the glycolysis and citric acid cycle can be taken out to make amino acids, right? So plants and bacteria can synthesize all 20, therefore you will find all 20 in these boxes, right? But for us, we can only synthesize about half. The others are required in the diet. All right, we must eat it, okay? So some examples, just highlight 3 examples for you, how these non-essential amino acids are being made. For example, glutamate over here we've already seen, right? We've already seen in the amino acid cycle, the glutamate, okay, can be formed from alpha-ketoglutarate. Transamination of alpha-ketoglutarate will give you glutamate. And from glutamate, this will act as a precursor to make glutamine, proline, and arginine. Structures are similar. Glutamate and glutamate especially, and then arginine. Alright, so this is possible, and this is the precursor molecule, the original molecule. And alanine and aspartate, on the other hand, they can be taken out from, we have also seen aspartate just now in the urea cycle reaction. Right, so if you connect it laterally, the oxaloacetate, right, combined with pyruvate and oxaloacetate transamination reaction will give you aspartate. Okay, so aspartate, and from aspartate, plants and bacteria can make all these other amino acids. Right, and from serine. Serine is the one with the OH group, right? Serine can be made from 3-phosphoglycerate. And from serine, you can make glycine and cysteine with a sulfur group. But in the mammals, this needs help from methionine. But methionine, we cannot make it, so we have to have a diet with methionine. Okay, in mammals, we need to have a diet with methionine in order for it to combine with serine, which can be made by the body, to make cysteine.